Hi, this video accompanies the Mitosis and Meiosis lab exercise, which is really just a bunch of questions about mitosis and meiosis as a review of the lecture material. But it also is requiring you to be able to visually recognize cells at different stages of mitosis on an onion root tip. So if you were in the lab in person, we would be using slides and you would be looking under your microscopes and learning how to visually identify the different stages of mitosis. Since we're not in the lab, this is the best we can do is show you a video of what you would be looking at. So if you first put that slide under the microscope, this is what you would see. You would see an onion root tip. Onion root tips, you imagine the root is rapidly dividing. So the cool thing about that is you can see cells at different stages of cell division as that root tip is dividing. So that the cells closer to the top are a little bit older. These at the tip are rapidly dividing and becoming bigger. So this is what you would see when you first put it under. So this is at 40x magnification. In other words, it's the 4x lens. Remember, the ocular is 10x. So if you use the 4x lens, you multiply it by 10 because of the ocular, and your total magnification then is 40 times the magnification. Now you can't really look at this and identify what stage of mitosis each of these cells are at, but it's really important to realize every single one of these is a separate little cell. Sometimes students look at this and they go, what stage is this at? Okay, well, every single one of these cells is at a different stage. Now, most of them are going to be at interphase because that's where the cell spends most of its time, is at interphase. So at any given time, you will see more cells at interphase than any other stage of the cell cycle. So you need to be able to identify interphase, prophase, metaphase, anaphase, and telophase. Then if we zoom in with our 40x lens in class, this is usually what you see. Sorry about this click to add text. I really don't want that there. Just gonna get rid of that right now. There we go. Okay, this is what you would see. Again, not super clear. I mean, I've done this a lot of times, so I can look on here and see, you know, this one's at metaphase and that one's at telophase. And it is challenging in the classroom because we do use the 40x lens, which gives us 400 times magnification. And you can start to see, you know, if there's not anything defined going on, if it's just a dark circle, then you will know that those cells are at interphase. But other than that, you know, it is hard to see the details. So we're going to zoom in even closer. Now, if we were in class, I would be able to come in and help you out and look through your microscope, and you would start to really see the pattern and you would get it. But since we are looking at this on a computer screen, I think this magnification isn't sufficient. So we're going to go one more step. We're going to use our 100x lens, which gives us 1,000 times magnification. And wow, now we can really see it. Okay, interphase. The chromosomes are uncondensed. The cell is just doing its cell job. The nuclear envelope is completely intact. So you won't see anything obvious going on. It's just going to be a dark, undefined nucleus. Okay, so we can look here, interphase, 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 interphase. All of these cells where you still see the nucleolus, which you don't need to worry about that if you didn't learn about the nucleolus in lecture, don't worry about it. But you can see it's a round nucleus, meaning the nuclear envelope is still intact. You cannot see any individual chromosomes. It's just a dark circle. When it starts to look, what I say is a little wormy, okay, then you know you're moving into prophase. Chromosomes are condensing. The nuclear envelope is breaking down and will eventually disappear. And so you're not seeing this nice round circle anymore. So look at this one. See how you can see the nuclear envelope is breaking down, starting to look a little wormy. In other words, the chromosomes are condensing and becoming visible. So this would be a prophase, this would be a prophase, this would be a prophase. Whenever you're starting to see individual chromosomes, and we've gone from this nice round nucleus to now we have something that's not so round anymore. So that would be prophase. In metaphase, the chromosomes have been pulled till they all reach the center with microtubules attached to each end of the cell. This would be metaphase. The onion root, the chromosomes have long chromatids. So it's not this nice little clean line, but nevertheless, they're all aligned in the middle. You can kind of see the microtubules here connecting those chromosomes to each end of the cell. So this would be a metaphase. This would also be a metaphase. You can see those really long chromatids there. This one up here, also a metaphase. Anaphase is when they pull apart and start moving to opposite ends of the cell. And honestly, there is not an anaphase in this view, but there is a telophase. Telophase is when we are dividing the cell into two new cells. And so the cell plate is how the plant cell is dividing the cytoplasm, starting to lay down new cell wall material between these two cells. And you can see new nuclear envelopes are starting to form. So we're getting two new nuclei and the cytoplasm is dividing. Cytokinesis is occurring by way of what we call a cell plate, since this is a plant cell. Let me show you another image so that I can show you what anaphase looks like, since anaphase is not shown on this particular view. And that's pretty typical. You know, you have to navigate the slide. You need to move around. If you were in the lab, you would need to move around the slide to find each stage. They're not going to just be bam right there when you first look. But they got lucky on this one. I can't imagine how long this person searched to find a view where all stages were represented all in one shot. Okay, so this would be an anaphase. You can see their chromatids have just pulled apart and have moved to opposite ends of the cell. So this is pretty late in anaphase. This would be pretty early in anaphase. They've kind of just pulled apart. They haven't reached the end yet. Metaphase, chromosomes are in a line in the middle of the cell. Okay, again, prophase, starting to see chromosomes. Nuclear envelope is not round and nicely intact. Telophase, this is very early telophase. You're starting to see that cell plate form and you're starting to see the nuclear envelopes reforming. I like to say in anaphase, it's like this. In telophase, it's like this. Anaphase, telophase. Move my hands. Telophase, anaphase, telophase. Okay. And then finally, ta-da, this also, you know, they took images and cut them and put them together. This didn't happen. And nobody found this on a slide altogether. Okay, this is awesome. Interphase, nuclear envelope completely intact, no individual chromosomes visible. Moving into prophase, you can see that nuclear envelope is broken down because we don't have that nice round nucleus. You can see individual chromosomes. Metaphase, they're all aligned in the center. And you see the microtubules, which is pretty awesome. Anaphase, they've pulled apart. They're moving to opposite poles, so this would be late anaphase. Even moving into kind of early telophase, but I can't really see a cell plate there. Then here would be the beginning of telophase. Cell plate is forming, nuclear envelopes are starting to reform. And ta-da, we've completed cytokinesis. We've completed what's called karyokinesis, which is division of the nucleus, and we have two new cells that will now, of course, go into interphase. So now each of these two new cells that just divided will start their cell cycle with interphase. So they'll move right into G1 at the end of telophase. Okay, that's it. Thanks.